So the process is called the rhizophagy cycle. Rhizophagy, root, phagy, eating, and cycle because it's a cyclic process, and I'll talk about that. And it's through this process that plants actually cultivate microbes and then extract nutrients from the microbes. And these microbes are endophytes. An endophyte is a fungus or a bacterium that goes inside plant tissues. And so mostly, if you're familiar with fescue grass, you know there's fungal endophytes in fescue grass. Okay, so this actually is an image that shows one of these fungi in fescue grass. But actually, there's all kinds of endophytes in all plants, fungi and bacteria. So every single plant has endophytes in it. And usually it's multiple endophytes, like small community of endophytes. Okay, and some of the features of these microbes in plants is that all plants have them, fungi and bacteria. Intracellular, that actually means they go inside the plant cells. And intercellular, they go between the plant cells. So they're both, in both cases. They will improve stress tolerance, abiotic stress tolerance, so they make the plants more resistant to, for example, drought, heat, heavy metals, anything that kind of relates to oxidative stress, they'll protect the plant from. So they're very important to be in plants for partially that stress protection. You have a weak plant without any endophytes. Weak, poorly developed. They suppress plant pathogenic fungi. Okay, we know that some of these endophytes will go out of the plant, go out into the soil, and colonize these fungi that are out there, these pathogens. And they don't necessarily kill those fungi, but they change the aggressiveness of those fungi. They'll make a fungus that's pathogenic, they'll turn it into a non-pathogen, simply saprophyte or even another endophyte in some cases. Exactly how they do that, we don't know. They will modulate plant development. They will actually control the development of plants, especially the roots. We see that kind of control by these microbes. They improve nutrient absorption, and this is one of the main processes that we'll talk about. So this is one of the nice things about doing work on plants is plants are all over the world, and so I get to travel to those places and collect and work on those plants. Okay, so this is actually the island of Bonaire and the Dutch Antilles, and they have these huge cacti. They call them Kadushi cactus, all over this desert island. And the fruits of these cacti are up here. But if you extract from those fruits the seeds, okay, that's shown here, and then germinate those seeds on, say, agarose, something clean, so that you don't have too many other contaminants coming in. You get a seedling that looks like this, and this is actually stained for reactive oxygen. This is stained, and you can see this orange root. Okay, so it's turning orange because of bacteria that are inside the plant. And what we do is we'll use this, I call it reactive oxygen staining, but it's a chemical that will turn brown if there's reactive oxygen. Reactive oxygen, hydrogen peroxide is one. Superoxide is a very powerful form of reactive oxygen. So the plants actually secrete superoxide onto these microbes. And so we're using the staining technique because that allows us to see those microbes. If we didn't have this, those microbes would be invisible. Okay, and it's because of that that people haven't discovered this process before, because nobody could see those microbes in the plants. But a few years ago we developed this technique, and you get this brown coloration using this technique. The stain, by the way, is a fancy diaminobenzidine tetrahydrochloride that we use. We just call it DAB. That's it. Okay, so this shows that root of the cactus, the Kadushi cactus. This is actually a root hair. These are root hairs here. And you can see inside. You can see all of these. Those are all bacterial endophytes. Those root hairs are loaded. Other root cells are loaded with these endophytes. And this shows a close-up. And you can see root hair. And you can see these divided pairs of bacteria there where they recently divided. Okay, so you can conclude that these microbes are replicating inside this plant. Okay, now they're actually not inside the cell itself. They're actually outside the membrane, the cell membrane, but they're underneath the cell wall. So this line there shows the cell wall. So they're really between the cell membrane of the root and the cell wall. And okay, so they get in that space called the periplasmic space between the membrane and the cell wall. In 2010, some investigators discovered that plants were internalizing microbes, yeasts and bacteria. And they actually they wrote a paper called Turning the Table, Plants Consume Microbes as a Source of Nutrients. And later they named this process rhizophagy, root eating. Okay, so what they did actually is they labeled their bacteria using a fluorescent protein, and then they visualized it. So you can see here the bacteria or the yeast, I don't remember which one this is, inside the cells. Here you see them inside the hairs. Okay, and so they actually went on to start a company where they're from Australia. They started a company where they're using yeasts as fertilizer. Okay, so they're trying to monetize that discovery. We discovered, actually in some work that has been going on over the past 8 years or so, we've discovered that the process is actually cyclic. And we published an article just a few months ago in the journal Microorganisms, Rhizophagy Cycle, an oxidative process in plants for nutrient extraction from symbiotic microbes. So we basically describe this process in more detail that we've been able to ascertain over these 8 years. And the process is like this. Okay, this is a diagram of a root. The microbes from the soil actually are attracted over to this root tip where these cells are dividing, and they grow in a zone out here, an exudate zone, just outside that meristem. And what happens is the bacteria or fungi become internalized into these root cells. And when they're internalized, they will lose their cell wall, and as they're hit with reactive oxygen, the superoxide that I talked about, the plant secreting. So that causes them to lose their cell walls. They form protoplasts. And then they will eventually trigger root hair formation. They trigger the hairs to form, because without the microbes, we don't get any hairs forming at all. And then they leave as they will exit the tip of these hairs as they elongate. They're actually, and I'll describe that process, they're actually ejected from the hairs by the plant. And they go back out into the soil, and then there they will acquire nutrients and carry those back to this exudate zone, and then some of them will re-enter again. So this cyclic process. So they acquire nutrients in the soil, nutrients are extracted inside the cells of the plant, of the root cells. Okay, and this little image, it shows a root hair, and you can see the bacteria there. There's fluorescent stained little cloud of bacteria coming out of that, being ejected out. This is a cell near the meristem, and you can see this is the cell here, the root cell. You can see the bacteria inside. So a lot of these microbes, this is a grass seed, this is a typical grass. I think this is a poa of some sort, bluegrass. These grasses will carry many of these microbes actually on the surface of the seed. They have lemmas and so forth. They have tissues that adhere to the seed. So they're adapted to vector some of these microbes are actually already in the embryo or inside the seed. So they have some surface, some interior. Different groups go different ways. But here you can see a germinating tall fescue seed. You can see the root coming out. See this kind of yellow around? These are the microbes that came off of that seed. So they're colonizing that seedling. They're colonizing at the shoot part and they're colonizing at the root part. So the initial colonization of the root is right at that meristem. And you can see these diagrams indicate this where the root exudates are. And so the microbes grow right there initially. Here another one shows those exudates there, and that's where the microbes grow. And this is an image that's an invasive reed grass, the giant Phragmites that's invasive in the United States. But this is the root. And here you can see the exudate zone here. And you see all these little dots. These are all the microbes. And what happens is the microbes enter right there, right where the cells are dividing, and they have thin walls, and the microbes go inside there. We don't really know how they go in. We don't know what process it is. We don't know. One theory is that the plant cells that engulf them. You know, there's another idea that the plant is manipulating the microbes and causing them to enter like a parasite would enter. So we don't really understand that process. All we do know is they do go in. So this is just a close-up of that. And you know, entry into these cells would be right in this area where you see this irregular surface there. The root tips actually show, even without the microbes, show high production of this reactive oxygen. This brown area shows where a lot of reactive oxygen is produced. We think that it secretes that all the time there. They're secreting the reactive oxygen, probably because as the microbes enter, they're going to be converted to protoplasts right away. They're going to be hit with reactive oxygen. Once they go in and they're hit with reactive oxygen, they lose their cell walls. Okay, and they form protoplasts. There's naked cells without cell walls. And there's actually, this shows an electron microscope image of some of these protoplasts of bacteria. And you can see these things just like bud out in little chains. And there's small ones, little tiny ones, and there's larger ones. So their rules of these cells aren't the same when they're in the protoplast stage as when they're in bacteria stage. Bacteria, you measure bacteria, they'll be very similar sizes, but with their cell walls. But with these protoplast stages, they can be really, really tiny, or they can be bigger, and there's highly variable. So that's an important difference. These also replicate very rapidly. So once they're inside and they lose their cell walls, the plant can replicate them and make lots of little copies and then send all those copies back out into the soil to get more nutrients. So this is very important for that in this rhizophaging process. Okay, some of the work I mentioned we did with giant reed grass. This actually is the root of giant reed grass. You can see this is a root cell here. And you can see these little blue or reddish brown structures. These are those protoplasts. And you can see there's a chain of them here. You can see this is the oldest one. You can see it's clear, larger and clear, and this smaller one is bluish. We stained it for protein using a protein stain that turns it blue. And so you can see these smaller ones have lots of protein in them. These older ones don't. What happens with reactive oxygen, it will go into the cell, make the membrane leak, cut up some proteins, and those proteins leak out, and so forth. So it's actually cell becomes leaky. And some of these older ones will just disappear, and you'll have only the younger ones that come out. So in order to make reactive oxygen, the plant actually uses oxygen. Takes oxygen from the atmosphere. And they use the enzyme. This is the enzyme actually in the membrane of the root. and they take that oxygen from the atmosphere and they make superoxide. So that's what's going on, just so you see a little bit of the chemistry. And this is hard to see, but this actually shows a lot of these little protoplasts inside a root cell. You can see big ones and then you see smaller ones in here, although it's very hard to see. So these cells get loaded with these protoplasts, microprotoplasts, and they degrade. Some of them degrade completely. Okay, this shows a little bit better. You can see younger ones here, they're blue staining. You see the older ones, they're bigger and clear. So many of them degrade completely. So they're just getting nutrients out of these. We used to think it was only bacteria that did this, but we discovered a couple of years ago or a year ago, something like that fungi also do this. And one plant that does it is this plant. This is snake cotton. It's amaranth family, and a number of species in the amaranth family will use fungi instead of or in addition to bacteria. And this is one of the fungi. It's a yeast called the black yeast. And these fungi will enter into the tissues at the root tip meristem also, and they will then turn into protoplasts again. And they form protoplasts that behave just like the bacterial protoplasts. They just bud like this inside in chains, many chains, and they go in the same place. They go into the periplasmic space in the root cell. Okay, so they're located in the same place. They call these mycosomes, though. You know, a fancy mycological term that, of course, mycologists who studied this had to give a special term for it. But it's just a protoplast. Just a protoplast. This shows it in that plant, in the snake cotton. This is a root parenchyma, and you see these protoplasts in here. This is the fungus. But you see also here, in the same root, you see these. These are bacteria. So the plant has both fungi and bacteria in it. So you have these communities. That's just visual evidence that you have more than one kind. Plants require these fungi and bacteria, these microbes, in order to develop properly. If you take them away, they don't develop right. They won't grow right. And for example, plants without these microbes don't show the root gravitropic response. Okay, so if there's no microbes there, roots will grow into the air, stick right up in the air, or they'll just grow on the surface. But they don't grow down into the soil. They just lie on the surface, perhaps waiting for the microbes to come. Also, roots without microbes, plants without microbes, don't show root hair elongation. They'll make roots, but no root hairs. Okay, so those microbes are necessary to develop root hairs on the plant. Okay, this shows a Bermuda grass seedling, and you can see the tip of the root here, and a little bit older part of the root here. And this is without microbes. All the microbes have been removed. There's no root hairs forming on these plants. Okay, with microbes, we put a pseudomoniad on it, which we got as an endophyte off of another plant. And we put it on, and you can see root hairs form immediately. And you can also tell maybe that there's this brown in here. That's where the microbes are located. Okay, and when they first go in, when they go into the root cell,
when you say immediately, like 1 day, 1 minute, 1 second, on the root hairs, like how fast is that time?
Days. It's a couple of days. In order, it goes in, and then the hairs begin to form, right? And they'll form over a period. It probably depends on the plant. So we're talking about maybe 2-3 days, something like that, before you get full elongation of the root hair.
Thank you.
You're welcome. Mike, wasn't it?
Yeah,
Scott.
Yeah, unless you're a bill collector.
Okay. I'm not a bill collector. Okay, so this actually what happens when they first go in, they have their cell walls. When they first go into the root cell at the meristem, they have this, that's the bacteria, the pseudomonads with the cell wall. And you can see in here some others. These are pseudomonads with the cell walls. But they immediately will lose those cell walls once they're hit with reactive oxygen. Okay, this is part of the root, and you can see, actually you look at this root, you see all these brown dots in there. Those are all the microbes inside those root cells. And you see these brown dots out here, those are all the microbes inside the root hairs. So there's lots of microbes in those roots. Okay, this is a close-up of a root hair, and you can see the microbe protoplasts actually in there, the brown staining. And that's from a Bermuda grass seedling. This is a Rumex crispus curly dock. Okay, we use that as one of our lab guinea pigs. And we isolated a Micrococcus luteus, that I mentioned it yesterday, from tomato, but it's distinctive because it forms these little tetrads of cells. And so you see, and the tetrads are distinctive enough, we can tell when they disappear. So when this is around the meristem, and you can see the tetrads that have just entered, and they're inside those root cells there, at least that outer layer of cells. This is without any microbes on it. You can see the same meristematic cells free of endophytes. This is a close-up of that. So you can see these tetrads are these little packets of bacterial cells. And they all still have their cell walls, but they're all inside those root cells. Immediately, they will lose the wall. They will lose their packets of cells. They'll separate out, and they'll form the little protoplasts. And here you can see them now. They're the same bacterium. But it's now forming little spherical structures. It's a close-up of that, and what you can see is that they're actually out in this periplasmic space. They're out here, just outside. There's the wall, there's a membrane there, and they're located outside of that, so you can see the position. Okay, just similar thing. You see the microbes, the protoplasts inside the hairs, protoplasts inside the hair there. Okay. Some of these will spontaneously turn back to walled cells inside the plant cells. So you see, here's our Micrococcus. This is from culture. Micrococcus luteus bacterium forming the packets. You can see most of these are little bitty dots there. Those are the protoplasts that are still in protoplast phase. But you do have some, like here, here, and here, that are reforming their cell walls and reforming their packets. So some of them spontaneously will go back. The plant will multiply the microbe in the root hair and increasing the numbers, and then eject it out into the soil. And this actually is a root of a grass, and it shows how these root hairs are elongating out away from the root surface. It would not be a good strategy to just put the microbes right there on the surface. It's better, more efficient to move them out into the soil and then squirt them out there. Yeah?
Quick question. You said that they populate inside of the root before they are exuded back out. Do you have any estimation in terms of the population increase inside of the root?
No, we really have no numbers. But we can see them replicating and I'll show you images that show that. And also the numbers that go in, if you consider entering one cell, you may have a dozen of them, but coming out, you have many, many more.
That was my question.
Yeah, you have many, many more. I can't tell you. It's likely what's going on is that the plant has good microbes, and it's replicating the good ones, and then pushing them out into the soil to populate the soil with the good ones so that they can go get nutrients for the plant. Right?
Exponential increase again.
So in that range, why do you think then some of these are rebuilding their cell walls within there? I mean, if they're
Yes, okay, that's
their cell walls, they're not able to go back out.
That's a very good question. That's exactly right. They can't go out. If they have cell walls inside, they can't go back out. Okay, so what's happening is Micrococcus luteus is very oxidatively resistant. And so it's able to resist the degradation that you'd be seeing like Pseudomonas Oxidatively resistant, what I mean is it makes antioxidants, right? And so the plant is squirting superoxide on it, it's making antioxidants, and so it's fighting that process. And it's, in a sense, it's not adapted, especially with this curly dock that we put in, it's not adapted to curly dock. And so it's not optimal association. Yeah. Okay, so the function of those root hairs is to put those microbes out into the soil. And this shows root hairs, and you can see this is fluorescent stain. You can see that all these clusters of microbes that are coming out. There's just spewing these microbes out of these hairs. You know, there's a lot that are actually coming out. This is a Poa annual grass, and you can see the little dots in here. Those are the protoplasts in there, and they are ejected out the tips of these, and you can see bacteria here reforming. These are protoplasts that came out. And you can see almost, it looks like a rod there, but they reform their cell walls once they're outside. So the plant actually feeds them out there. It gives more nutrients so that they can increase in size.
What is it feeding them?
It must be sugars. It must be sugars. I don't know exactly. I'll show you some images of yeast that does it. You'll see that they increase. So, I mean, nutrients, in fact, must be going out of the hairs.
That was my question.
Yeah.
The oxide that they're putting out there, that
It's not going
it's
out.
not an attractant, it's not an
No,
attractant,
no. And as soon as they go out of the root hair, they're no longer exposed to superoxide. They're no longer exposed to that. They escape from that, and then the walls reform and they go out. They get flagella and so forth. So this actually shows tomato root hair tip. You can see these microbes in it. You can see a stain for reactive oxygen. See they're all brown, so they're getting hit with reactive oxygen there. And you see on the surface, you see some little rods forming. Okay, so the model, the way that we think this works is, there is cyclosis, circulating cytoplasm here that goes around like this, goes all around this cell, constantly circulating. And that drags these microbes along this rigid wall here. And this tip is thin walled, very elastic, and it just deposits them there at that tip, and they accumulate there. And then they stimulate as this protoplast, this root hair protoplast, pushes, elongates more, and pushes them, puts pressure on them, and then they squirt out little pores that form there as protoplasts. The protoplasts squeeze out the pores. Then they reform their walls. Okay, so we'll show, I have some images that we'll look at that. This is a little sedge, again from bone air. It occurs in these limestone rocks. There's hardly any soil there, but it's a cool plant to work on. Okay, this actually is that sedge root hair. And you can see the root hair right there. You can see in here the microbes out there. Right there. And I'll show you another image that shows that movement of the cytoplasm like that, dragging those microbes around. Dragging them around, roughing them up, causing them to split apart and make smaller protoplasts. And okay, so here this shows actually some replication in those hairs. See the hair? You can see the protoplast here, the different sizes, smaller ones there. Okay, so those clusters are moving around, breaking up, and so forth. Okay, this is that sedge hair. You can see it's swollen at the tip. You can see all those protoplasts in it. You see protoplasts here. Actually here, their walled cells are coming out. The bacteria are coming out of that in a mass right there from that tip. They're being pressed out, pushed out. Yeah?
Is it physiologically possible to translocate through the plant into the leaves from the root area?
These bacteria go throughout the plant, and they're seed transmitted, so they actually are also colonizing that ovary tissue, so that when that seed is fully formed, it has some of these microbes on it. So yes, you know, how significant, I mean, it's obviously significant. Which ones do some not do that? Yes, some don't, probably don't do that. But some are seed transmitted, so they're going through the plant.
Follow-up question. Would it be possible that you could have a Shiga toxin producing strain of E. coli enter a plant through the root?
Certainly, you could have human pathogens that go into plants. You could have. But, you know, one thing is when we eat it, you know, we probably eat lots of potentially pathogenic microbes, but they go in the gut and you have a diverse community, and that community in the human gut and in animal guts polices themselves mostly, so they suppress virulence of many of those microbes.
I guess what I was referring to is some of the romaine E. coli outbreaks. If pulling the E. coli out of the soil and pushing into the vegetative portion of the plant.
It is possible, yeah.
It's possible.
If they go in, it's possible that microbes can go throughout the plant, even pathogenic microbes. It is possible. Okay, so this is one movie of that sedge root hair, and what you see is the root hair here, and you can see the microbes here, clusters of microbes moving around, moving like this, but you can also see the tip where those clusters of microbes are locating. And so they're being jumbled around, and as that protoplast here that goes into there elongates more, it pushes them through little pores there at the tip.
Is that real time or is that time lapse?
This is very close. This is probably double speed, but yeah, some of these are moving pretty fast. I mean, we've estimated a time 8 to 11 micrometers per second. So that's pretty fast, really. So how do we do this, right? How do we do this? We grew these in agarose, these plants in agarose, and we turned the plate upside down to visualize it with the microscope, and then we did the photography that way. It's very close to real time. You can visually see them moving. So it's not really time lapse. Let's speed it up a little bit.
So I understand circulatory systems within me. I'm no expert, but you know, I understand I have a heart that provides the fluidic force to circulate. Within a plant, what is actually causing the motion of fluid?
So plant cells have an internal cytoskeleton. So they will use that cytoskeleton. It's made of filaments and tubules and things, and so they can move those, short them, make them long, and to make the cytoplasm like that move. So this is a circulatory system in plants, and you know, we can guess why they move like that. Why? I mean, the function here is probably to drag those microbes to the tip so it can shoot them out, but it also may be to get nutrient exchange between the microbes and the plant, because you don't have a gradient developing there. You're constantly breaking that down so it's the fastest possible transfer. Okay, so that may be the function. We're guessing at it, but it's logical.
And you're also having two-way traffic within the same tubule.
You can have two-way traffic. You can have two-way traffic. Yeah. Typically, you know, I describe here where it's, I mean, you can see this. I mean, here's another one where a pore is going to form here, and it looks like you have multiple traffic, multiple ways.
And not random, it's linear.
Say it again?
It's random. It's not random.
It's not random. It's not random. Yeah, it's not random. No, plants are doing this. Plants are really doing this. We can see it. And they're ejected through pores. You know, I don't know that we see an ejection right here, but you see a pore forming. So these pores form and they go out. Okay, this actually is a fescue grass, and you can see the root hairs here. You can see all this. This is sped up a bit. You know, so it's really cooking. This one's really cooking. But you can see a protoplast being squirted out of one of those. At least we think that's what we're seeing there. We're trying to get better and better images. I have an undergraduate working on this now. We think that what happens with the root hairs is they will grow to until all the microbes are gone, and then they stop growing. They'll just stop. Okay, if there are fewer microbes, you have short root hairs. If there are many microbes, you get big root hairs. And we think what's going on is at that tip, the microbes are secreting nitrous oxide, or it could even be ethylene, but most likely it's nitrous oxide. And that nitrous oxide stimulates the root hair to elongate. So as long as the microbe is there, nitric oxide is formed, and the hair elongates like that. Okay, so that's what we think. At least that may be the chemical mechanism. Okay, so tomato, look at tomato, development of tomato, these root hairs, these root cells will form these little papillae on them. Okay, that's the precursor to the root hair tip, actually, precursor to the tip. And those papillae then will accumulate the microbes. You can see a papilla there, and you can see the microbes in it. See, there's not very many there to begin with. Okay, the papillae enlarges, you can see more microbes in there. And here you see the microbes there. You see here's an older root hair of tomato, and you can see how many microbes now are at the tip, at that papilla, at the very tip. And of course the wall here is very thin and elastic. And it's breaking, as we said, making little pores in it, expanding. And this shows the tomato again. You can see how expanded it is with all these microbes in it. And there you see another one. And there you see another one, microbes there. Here you see the expulsion of protoplasts from the tip. You can see those protoplasts accumulating out there. Okay, this is tomato again, and you can see all those microbes there. You can see the microbes on the outside, and some just almost being extruded there. English ivy, these have roots that go into the air, so it's easy to see them. These are some of the roots, some of the air roots, but you can see here from English ivy, this is a root hair. You can see microbes there at the tip, and you can see the little pores right there forming there. It's a close-up of that. You can see those little pores that develop, and the microbes are the protoplast get squirted through that. This shows English ivy hair again, and you can see the protoplasts accumulating on the surface there as they're pushed out. Yeah.
I have a question.
Yeah.
As they're expelled out, is there something that they absorb from this, the root surface to attract certain metals? Like, for example, if there's like a zinc deficiency, do they have certain things on the surface to chelate that?
Yeah, we'll talk about that. Yes. So we've been putting thought into what the mechanism, how they might be working, and I'll go into that very shortly, and how we think that's happening. Okay, just shows more protoplastic tip, and this just shows more of that. You know, to get the point. You can see the pores there. You see the bacteria reforming their cell walls here. And you can see they're in tetrads here, and they just spill off that tip. You can also see the channels through the cell wall there at the tip, and the bacteria reforming right there. And underneath these spherical protoplasts. Okay, you just show the spilling off there. Okay, and this is actually the fungus when they come out. This is a fungus, a yeast again. Rototorula is its name. But you see the protoplast inside, and this is actually in clover. This is clover, so clover does it too. And you see those protoplasts, you can see protoplasts there. You can see them coming out. You can see the youngest ones are smaller. Oldest ones out here are bigger. And so what has to be happening is here, these yeasts are getting nutrients as they're being produced outside, suggesting that the flow of nutrients actually is out of those hairs, going to the microbes as they reform there. And what nutrients? Carbohydrates probably. That's what's cheap for the plant. It can make lots of photosynthate. Okay, this just shows that again. In clover again, you see the formation of the... Okay, nutrient absorption function, how it works. Yes, Scott?
So the root hairs, are they ever pulling in?
We don't
from the
know.
soil, or is it only?
We don't know. We don't know the answer. We can't say really. One thing is for sure, root hairs die very shortly after all these microbes are ejected. So do they really absorb micronutrients from the soil? We don't know. We don't know. I can't say no, but it certainly looks like their function is to put the microbes in the soil rather than absorb nutrients from the microbes from the soil.
Can we get a refund on our textbooks?
I know, me too. All the way back to like 2nd grade. Yeah, if it's true. But okay, so to test this, we did a nutrient, to test to see how these microbes were assisting in nutrient absorption, we did an isotope tracking experiment. This is actually where we labeled some protein with isotopic protein, nitrogen 15 it's called, so we could track it, and as it goes into the plant. And basically we put the protein, labeled protein, into agarose here, mixed it in there, and then we put the plants with microbes here, bacteria abundant, okay, and without over here. And then we measured the amount of isotopic nitrogen that went into each of these seedlings. And what we saw when we did that is that when the microbes are present, you get this much. When the microbes are absent, you get this much. And so that is about a 30% difference. So it looks like in terms of nitrogen, the microbes are providing 30%, they're worth 30% of the nitrogen that the plant gets. That's not 100%, but it's a significant increase in nitrogen. Okay, so this is a plant grown with all the microbes present, and you can see it's a nice green plant, they're big, lots of roots. And then when we take the microbes away, you see now we've got yellowed plants, perhaps an iron deficiency issue, and hardly any root development there with no microbes. Then when we put on one of our endophytic bacteria, we reacquire, look at root development here, and they're green again. Okay, here also we have a bacillus, an endophyte that took off of these plants, and then we put them back onto sterilized plants. And you can see that the bacillus also is reacquiring that growth, the green again, chlorophyll, lots of root development. Okay, so how do microbes do this? Okay, so microbes have structures called siderophores. They have some other, these are actually iron molecules, this is how they get iron. Cell walls will absorb metals to them. They also will actually work with plants and getting microbes from the organic acid metal complexes using special transporters that they have, and I'll show that process. Microbes also will take nutrients from other microbes. Okay, and how do they do that? They make what are called biosurfactants. And those biosurfactants make membranes leak. So a microbe like, for example, bacillus will produce surfactant and make membranes of other bacteria and fungi leaky, and then they can then acquire those nutrients. So there's, we think that's part of this phenomenon. Siderophores, okay, how do they work? There's a special molecule here that microbes produce, this little blue thing here, that's the siderophore. Here they put that out, send that out, it attaches to an iron, for example, and then it will carry that back and get into the receptor, and then the microbe will absorb it in. Okay, the cell walls of microbes have a negative charge, and the iron will just stick on it. Metals will stick on it. Zinc will just stick on it. This actually is a bacterium, and it shows all these little silver dots. That's silver particles sticking to the wall. So when the plant takes the wall away, it gets microbes right there. It carries microbes. Okay, the other process is we know, and I think this is getting to the question that you asked a few minutes ago. We know that plants secrete organic acids into the soil, citrate, acetic acid, for example, citric acid, malic acid. And we also know that those organic acids will go out and attach to metals, iron, zinc, and so forth, attach to those metals and complex, and just cover them over. Microbes have these organic acid metal complex transporters. So the microbes can go find those complexed metals and then hook onto them and suck them in. So, and then once they get them, they can then travel back to the plant and re-enter the plant where it can be harvested. So this is a kind of a system where the plant is actively working with the microbe to maximize nutrient absorption. Okay, and this just shows details of some of that. The microbe can use the citrate as an energy source, so it actually is rewarded for this, for doing that. All right, so I'm not going to go into that. I already mentioned that. What happens to plants without the rhizophagy cycle? Okay, so this is a rice, and we removed all the microbes and we put a pseudomonad in it. And you can see these big seedlings. Okay, and these have no microbes at all, and you can see very tiny seedling development. A little bit older seedlings, you can see here. Here, this is with the microbes, with the Pseudomonas. You can see the plants are bigger, maybe about 30% bigger. And you also see the soil adhering here. So the relationship with the soil, the root hairs and that relationship sticking the soil to the roots is more significant here than you have here without the microbes. So it does appear to be significant. Okay, the very last thing I'm going to mention is that the rhizophagy cycle may be affected by carbon dioxide levels. And the reason for that is that carbon dioxide in the air will suppress reactive oxygen in the plant. And I'm sure you've all heard that there's some work going on looking at nutrients like zinc and iron in crops, decreasing in these carbon dioxide enrichment experiments where they expose plants to carbon dioxide and then measure nutrients going into the plant. So it's suppressing those nutrients. And that's a few years now people have been looking at that. Well, as it turns out, we did some experiments to evaluate, whether high CO2 is impacting the reactive oxygen. And this just shows, this is at current levels of CO2 in the air, and you see it's a bit darker. This is in an experiment with a bit higher. This is like the 50 to 100 year level, so it's a little less, so the levels of reactive oxygen appear less. And we actually, to measure this more precisely, what we did is we can grow plants, remove all the microbes, grow the plants in protein, and protein will stimulate reactive oxygen formation. And then we do that different CO2 level experiments. And then stain it for reactive oxygen. You can see all this brown, roots are in there. You can see at the current levels of CO2 in the atmosphere, you see it's kind of thick, broad zones around those root tips. But this is in the higher level, the 50 to 100 level. You see they're very fine, very fine. There's less reactive oxygen around those tips. So we don't know if that means, that plants, that this is just destructive, this climate change is going to be destructive to the rhizophagy cycle. Plants might be able to adapt. We just don't know that yet. And we don't know, this actually has to be confirmed somewhat. What is clear is that we want to conserve this underground biology that's happening with these microbes. And what that means is, for one thing, they're on seeds, so don't use seed treatments. Anything that sterilizes the seed or interferes with microbes on the seeds, don't remove those. Organic amendments to the soil, increasing organic matter is important. No-till, don't disrupt, don't churn the soil so that you disrupt those microbial community, and so forth. So that's really all I have to say. Right? Any other questions? I saw a hand.
I had a question. So when you mentioned that microbes absorb the metals on their cell wall, do the plants get those nutrients after the cell wall sloughs off from the reactive oxygen?
I am sure they do, but it's logical that they do. But to do the experiments where we label the cell wall and then track the metal into the plant cell, we haven't done those experiments yet. Come on.
Quick question for you is two quick questions. One is, do you think this could be really what's driving epigenetic effects is the protoplasts? And then the other question Go ahead and answer.
I do. I think that this is one of the unknown determinants of things like stress tolerance and growth, things that are not necessarily genetic. So this is something that when you breed the plant, you're not really looking at, but it has a major effect on plant growth and hardiness and so forth. Yeah.
And then the second question is, when you do the math on the amount of trace minerals that a 22 ounce or 1 pound per acre glyphosate application would make, it does not correspond to the typically decrease in the total tissue content that we see in the plants. If you spray 1 pound per acre of active ingredient, the total chelation ability of that is nowhere close to the total nutrients per acre that we're taking out of tissue. Is it possible, because that is also an antibiotic, patented as an antibiotic, that within plant we are destroying the protoplasts, which is the engine for the uptake of the micronutrients? So we're seeing that decrease is not because of glyphosate tie-up, but because of lack of microbial acquisition after application.
Monty, I have not even thought about that. So I don't know. There's a lot of, ramifications for this. And I guess what will glyphosate have on the rhizophagy cyclic, right, for an experiment, for example. And so that's something that will be worth looking at. That we'll try. We're trying to see what affects the rhizophagy cycle now, so everything that affects it, we kind of want to know, to learn everything about it that we can learn. And so an experiment like that is the kind of thing that we would try to follow up on. Yeah, thank you for asking it.
As it pertains to the life cycle of that root hair, is it better to overstimulate where it pumps a huge amount of microbes out, or is it better to keep it alive longer?
We don't know. They don't live very long, those hairs, though. Like that. Yeah. We can actually get them to live longer if we put them in protein. Maybe they're functional more because they're surrounded by protein continuously. But they die after just a couple of days if you don't have protein there. That is after the microbes have been secreted. The other thing is that, most roots, older parts of roots, don't have hairs on them at all. So they slough off. The hairs are really a phenomenon of that tip, right behind the tip. Yeah, Monty.
How much, when you've observed the division and the replication of the protoplast within the root, how much genetic or gene transfer is there between protoplasts and between plant and protoplast? Is that observable?
No. I can tell you something though. I mean, it's not a crazy idea. Some of these bacteria, the alpha proteobacteria, will go inside the cytoplasm. And some of them will actually, and we can see them in there, they go into the nucleus. They go into the nucleus itself. And then we think that they do that so that they don't get degraded because they're oxidatively degraded in the cytoplasm. And so they can escape that by going into the cell's nucleus. We don't talk about that much because we haven't done a lot of work on it, but we can see them going in. And anyways, yeah.
Can you explain to me, it is a really basic question, but it helps me build my foundation. What's the difference between oxide and superoxide, and how does that seem to strip the cell wall and it becomes a protoplast? What's actually the chemistry mechanism?
That's a good question. Okay, so we hypothesize that the reason that there is a difference. Oxides are like aluminum oxide or something, right? Another compound that has an oxide component to it. Superoxide is oxygen itself that's charged with an extra electron so that it can zap directly. And so it's very powerful superoxide. Hydroxyls also are very powerful oxidants. Okay, so reason we think and we don't know for certain how this is working, but the superoxide can, for one thing, degrade the wall itself, and also, because it goes into the membrane, it can degrade those proteins that form the cell walls in the membrane, and so when those are damaged, they can't make new walls. So as long as the superoxide is there, so that's what we think. But there's lots of unknown, and that all has to be experimentally evaluated, right? We can see what's happening, and we know that they're losing their walls because they're losing their shapes. And this phenomenon of protoplast formation is very well known in bacteria, human and plant tissue. So this protoplast formation is not new. Did that answer the question? Mm-hmm. Yeah. Okay. Yeah.
You had mentioned not to use seed treatment. What are the disadvantages of that?
Well, okay, so the reason I say that is because you don't want to remove the microbes that are naturally present on the seed. Those microbes on the seed give the seedling a kick, critical to seedling development, right, and protection from disease. I know that some seed treatments will, for example, we have some Bermuda grass where they remove the husk of the Bermuda grass and they replace that with fungicidal coating. Well, in removing the husk, all the natural microbes are gone. And of course, it's necessary now to put fungicide on the seed because it's so vulnerable to disease now that we've done that. And of course, there's reasons people do coatings, and I understand it's very common now. A coating, for example, I would think a coating would be good that would enhance the microbes that naturally are carried on the seed. So you give them a boost and out into the soil. You kind of send the plant out, send the seed out with a lot of its microbial subservience, microbial slaves, so to speak. Yes, Scott.
So there's a popular practice of putting Mycorrhizae in with rice seed soaking. So the rice seed is soaked, someone's flown on it, it drops down through the air into the water. And then as they're soaking, they're adding Mycorrhizae to the soak water. And my question is it doing any, just for you with your knowledge, I know you haven't experimented.
Well, I do know.
Do you think it will take, it does help? How long would it take?
Mycorrhizae do help. The Mycorrhizae don't help all plants. I mean, I would say what you would need to do is to just do a little experiment yourself and evaluate if you see an effect there. Is it really helping with the rice?
Right, but yeah, no, like it helps with rice. That's proven, but I'm thinking if they're wasting their time putting it in water, putting it in for a day, it'd be better if it was in the soil than just being put on a seed. Is the seed picking up Mycorrhizae in a water solution for a day? The hyphae might be coated onto the surface of the seed. That's conceivable. Now, does it really work? I can't even tell you if that. I can't tell you if that really works. We haven't looked at that. And I mean, conceivably, yes, you could put hyphae on the seed surface or vesicles, spores of the Mycorrhizae that could adhere to the rice. Okay, so it's theoretically possible, but does it really work? I don't know. Another question.
Okay.
So is Protoplast, does it leave the root here? What's going out? What is being released?
Protoplasts.
Okay.
And it's necessary because those pores are little.
Right.
And if they get their walls, they're trapped in there. So responding properly and forming protoplasts is very important. It has to be a good connection, right? They have to be matched so that the protoplast form and it can replicate and then they get squirted out and put massive numbers back into the soil. So that's really important.
So, but are they released on a mission? In other words, are they released being told by the plant, go get me iron?
Well, so they naturally will go get iron, right? I mean, the plant is
Will they go get
secreting.
anything that they run across?
Yes,
Or
yes, they need all that stuff. They need, they're going to get everything. They're going, so the plants are manipulating these microbes, right?
That's
The
what I'm
plants are
asking.
farming these microbes. That's what they're doing. I mean, I'd say it's like, looking at the system, plants appear to be in control of this whole process. You know, I don't know, it's not Russian science, or the old, you always, in National Enquirer, you know, where they were, you hear plant thinking and stuff like that. Nothing like that, but it's obvious that the plants are in control of this whole process.
I think to follow up on Scott's question, you and I were discussing that, and you said there's several species that it has to choose from, and it will send out the species and propagate the species
It does.
that correspond to the nutrient that it needs.
And I think that's automatic too. I think that's automatic because the ones that enter are compatible. And if they respond by producing protoplasts early and get replicated and don't reform walls inside, then they go squirted out to the soil. So I think it's like an automated process, self-selecting to select the microbes and to increase those microbes in the soil. The microbes that they select probably are also the best microbes for getting nutrients. Things like Bacillus or Pseudomonas, you know, they have abilities to get these nutrients. That they also respond back and come back to the plant. So many of them are swimmers, so they swim and can go far, ranging far to get nutrients, then come back for more photosynthate.
I think it's time for us to get squirted out of
I
root
think
tip.
so too. I was just
Good
gonna
food.
say.